Hey guys, it's Adam here. I'm just going to be talking about the DNA replication for today. This is essential for biology 1101 because, and biology as a whole, it just helps you understand and set a basis for transcription, translation, and future chapters ahead. As a brief overview of what we'll be talking about here, it's important to note that DNA replication as a whole is the production of two DNA molecules that are identical from one DNA molecule. It's the basis for biological inheritance, where organisms transfer genes to their offspring, and also occurs in all living organisms. As many of you may already know, the monomer of a molecule in a chromatid of DNA consists of a pentose sugar with one hydroxyl group coming off one end and a phosphate head coming off the other. Notice how the hydroxyl end is called the 3' end and the end with the phosphate head is called the 5' end. The pentose sugar with one hydroxyl molecule coming off of it is called a deoxyribose sugar because it only has one hydroxyl group coming off of it. Coming off the side of each of these deoxyribose sugars is a nitrogenous base. The resulting structure is double helix shape where each backbones are polymers of the monomer we just drew. This double helix shape is a semi-conservative model, and the process of DNA replication is called semi-conservative replication. So through a basic 5-step process, this double helix structure is splitting in half, whereby each strand is replicated to, again, create two identical DNA molecules. The first step in DNA replication is the role of DNA helicase. This is an enzyme that binds to our DNA structure and breaks apart the hydrogen bonds between the nitrogenous bases. By separating the two strands into single strands while moving in one direction, a replication fork is created. This is the point at which both strands are separated by the helicase. Next, an enzyme called RNA primase, or just primase, produces short RNA primer strands by attaching bases that code for RNA sequences. These temporary strands provide a foundation for the next enzyme, DNA polymerase III, to bind to, as it can only bind to a double-stranded structure. Because the RNA primer strands slightly peel off the DNA strands, the DNA polymerase III is able to bind to the end of the RNA primer strands, and it adds the corresponding bases to the template strand. Please remember and take note that for coding, adenine bonds to thymine and guanine bonds to cytosine. Also remember that DNA strands are read from the 3' end to the 5' end, but are built from the 5' end to the 3' end. Think of DNA polymerase III as a Wile E. Coyote cartoon on a set of train tracks gliding along. He realizes that half of the track ahead isn't filled, so he grabs pieces for a new railing, or in this case, just a bunch of nitrogenous bases, and begins sliding them in right in front of him. This occurs in a continuous process on the strand that reads from 3 to 5 towards the replication fork, but happens in a discontinuous fashion when on the other strand that builds away from the fork. The fourth step in DNA replication includes the enzyme DNA polymerase I that replaces the RNA primer nucleotides with the DNA nucleotides. The primer nucleotides are the strands that were used by the DNA polymerase III to start on its new track. So it gets rid of the temporary RNA track and replaces it with a shiny new DNA track. The last step in DNA replication is where the DNA ligase joins the DNA fragments together, or so-called sealing the cracks in the track. It does this by reinforcing the phosphodiester bonds in between each of the monomers. Going back to our structure here, this is when this enzyme reinforces the bonds in between the hydroxyl molecule on the bottom and the phosphate head on the top of the below molecule, just like this. So there you have it. This replicated strand over here would also happen on the other strand, and therefore you would end up with two complete DNA molecules. Here you could see the two strands of DNA are produced by the creation of the leading strand and the lagging strand. Make sure you review the 5 steps that appear here. Remember, it starts with helicase, unzipping the double helix, followed by the RNA primer, making temporary strands for the DNA polymerase to use to create and attach new bases for the new strands. This is followed by the DNA polymerase 1, replacing the RNA primer strands with DNA segments, and lastly, the DNA ligase, which is used to seal each of the new strands together. For review, I've listed the corresponding base pairings that DNA polymerase 3 and 1 abide by when building off of template strands. These are adenine pairing with thymine and guanine bonding with cytosine. If you wanted to challenge yourself, try looking back in this video just after the helicase splits the DNA and try to come up with the corresponding strands for the template strands. If you need more practice, try pairing the correct bases with a random assortment of letters, including A, G, C, and T, in a straight line to mimic the matching pairs. Before I sign off, just know that I pasted a couple links here to some online quizzes from Crash Course Biology and Khan Academy if you wanted to do some more research on this.